All right, everyone, get ready because we're diving deep into your Class 12 biology textbook today.
That's right. We're tackling molecular basis of inheritance. It's a pretty dense chapter.
It is, but we're going to break it down into manageable pieces so you can not just understand it, but actually ace those exams.
We'll be highlighting the key concepts, the aha moments that make this stuff click.
Think of this as your ultimate study guide, your secret weapon for conquering this chapter.
So let's start at the beginning with a molecule that holds the blueprint of life, DNA.
DNA.
Yeah, everyone's heard of it, but it's way more than just a fancy acronym.
Right. It's a long molecule made up of these building blocks called nucleotides.
Nucleotides.
Mm.
Okay, I vaguely remember those from class.
Well, each nucleotide has three parts: a sugar, a phosphate, and a nitrogenous base.
And there are 4 different bases, right?
Adenine, thymine, guanine, and cytosine.
Exactly. And they pair up in a very specific way. Adenine always with thymine and guanine always with cytosine.
Like puzzle pieces fitting together.
Precisely. And that pairing creates the famous double helix structure, the twisted ladder shape that we all know and love.
Okay, so DNA structure 101. But why is it so important?
Well, think about this. If you unraveled all the DNA in a single human cell, it would stretch over 6 feet long.
Wait, 6 feet? That's taller than me.
Yeah.
How does all that fit into a tiny cell nucleus?
That's the magic of DNA packaging.
Packaging.
Yeah, picture this DNA wraps around proteins called histones, kind of like thread on a spool.
Okay, I'm visualizing that.
These little bundles of DNA and histones are called nucleosome.
Nucleosomes, got it.
And then those nucleosomes get all coiled up tighter and tighter, ultimately forming chromosomes.
So it's like a super organized filing system for all that genetic information.
Exactly. It's incredibly efficient, both for space saving and for controlling which genes are active.
Now, I remember learning that DNA was discovered way back in the 1800s.
It was. But it took quite a while for scientists to figure out that it was the actual genetic material, the carrier of heredity.
Yeah, I remember reading about those experiments. It's like a scientific detective story.
It is. It started with Griffith's experiment in 1928. He was working with bacteria and found this mysterious transforming principle that could turn harmless bacteria into deadly ones.
So something was changing the bacteria's genetic makeup?
Exactly. Then in 1944, Avery, McLeod, and McCarty took center stage and showed that this transforming principle was DNA.
Wow. So they finally cracked the case.
Well, not quite. Some scientists were still skeptical. It wasn't until the Hershey-Chase experiment in 1952 that the evidence became irrefutable.
What did they do?
They used these tiny viruses called bacteriophages, which infect bacteria.
Bacteriophages.
They labeled the DNA and the protein coat of the virus with different radioactive markers. And guess what? Only the radioactive DNA made its way into the bacteria, proving that DNA was carrying the genetic instructions.
Case closed, DNA is the culprit.
Precisely. But you might be wondering why DNA? Why not RNA?
Yeah, they seem pretty similar, at least from what I remember.
They are related, but DNA has some key advantages that make it ideal for storing genetic information.
Like what?
Well, first, its sugar deoxyribose is more stable than RNA's ribose sugar.
So DNA is built to last.
Exactly. Plus, DNA uses thymine, which is more chemically stable than uracil found in RNA.
Okay, makes sense. And I guess that double-stranded structure helps too, right?
Absolutely. It's like having a backup copy and a built-in error checking system.
So DNA is the ultimate safe for genetic information. Wait, didn't RNA come first?
That's a fascinating idea called the RNA world hypothesis.
Oh yeah, I've heard of that.
RNA is more versatile. It can both store genetic information and act as a catalyst, like an enzyme.
So it could have been the original player in the game of life.
Precisely, with DNA evolving later as a more stable way to store the genetic blueprint.
Okay, so we've got our genetic blueprint safely stored in DNA. But how does a cell make copies of this massive instruction manual?
That's where DNA replication comes in. And you know who actually predicted how it would work?
Who?
Watson and Crick, the double helix heroes.
Really? Just by looking at the DNA structure?
Yep. They proposed a semi-conservative replication model, meaning each new DNA molecule would have one old strand and one newly synthesized strand.
So it's like a copy-paste, but with molecular magic.
Exactly. And their prediction was confirmed in 1958 by the Mendelssohn-Stahl experiment.
Using heavy nitrogen isotopes.
Precisely. They showed that DNA replication is incredibly precise, ensuring that the genetic information is passed on faithfully.
Okay, so we've got the blueprint duplicated, but how does this information actually get translated into building the proteins that make us Well, us.
That's where transcription comes in. It's
Sure.
like transcribing a recipe from a cookbook, but in
Okay.
this case, we're copying DNA information into RNA.
So RNA is like the working copy of the recipe.
Exactly. And the key player here is RNA polymerase, an enzyme that binds to DNA and creates an RNA transcript.
Okay, and what tells RNA polymerase where to start and stop?
Good question. There are specific regions on the DNA called promoters and terminators.
Promoters are like start signals and terminators are like stop signs.
Precisely. And the DNA sequence between a promoter and a terminator is called a transcription unit. It's like transcribing just one chapter of the DNA book.
Got it. But is all RNA the same?
Nope. There are different types, each with its own specific role.
Like what?
Well, there's messenger RNA or mRNA, which carries the genetic code from DNA to the ribosomes.
Ribosomes, those are the protein-making factories of the cell, right?
Exactly. Then there's transfer RNA or tRNA, which brings the amino acids, the building blocks of proteins, to the ribosomes.
Okay, and what about ribosomal RNA?
That's rRNA. It's a structural component of ribosomes, helping them do their jobs.
So it's like a whole team of RNAs working together to make proteins.
Precisely. But things get a bit more complicated in eukaryotic cells like ours.
How so?
Well, in bacteria, the RNA transcript is ready to go right after transcription. But in eukaryotes, it needs some extra processing before it can leave the nucleus.
Processing. What kind of processing?
First, the initial mRNA transcript called pre-mRNA goes through splicing, where non-coding regions called introns are removed.
Introns, what are those?
They're sequences that don't code for proteins. They're like unnecessary bits in the recipe that need to be edited out.
So you're left with only the protein coding sequences, the exons?
Exactly. And then a cap is added to the beginning of the mRNA and a tail to the end.
Why is that?
It helps protect the mRNA and guide it to the ribosomes for translation.
So it's a complex journey for mRNA from transcription to finally reaching the protein synthesis stage.
It is. But now here's the big question. How do we go from the language of nucleotides to the language of proteins?
Yeah, how do those 4 bases ATGC code for 20 different amino acids?
That's where the genetic code comes in. It's like a secret decoder ring that reveals the relationship between nucleotides and amino acids.
So how does it work?
Well, scientists figured out that 3 bases together called a codon specify one amino acid.
A codon, a 3-letter word in the language of DNA and RNA.
Exactly. And there are 64 possible codons, which is more than enough to code for all 20 amino acids, and even have some extra codons for start and stop signals.
Okay, so it's like a dictionary that translates the language of DNA and RNA into the language of proteins.
Precisely. And scientists painstakingly figured out which codon corresponds to which amino acid.
Wow, that must have been a lot of work. But now we have this amazing tool to understand how genes are translated into proteins.
We do. And there's another molecule that plays a crucial role in this process, transfer RNA or tRNA.
tRNA.
Right, we talked about that briefly.
Well, tRNA is like the interpreter. It bridges the gap between the mRNA code and the amino acids.
How so?
tRNA has this cloverleaf-like structure with one end that can bind to a specific codon on mRNA and another end that carries a specific amino acid.
So each tRNA molecule is like a matchmaker, bringing the right amino acid to the right spot on the mRNA template.
Exactly. And here's something interesting. The genetic code is actually degenerate.
Degenerate, what does that mean?
It means that multiple codons can code for the same amino acid.
So there's some redundancy in the code.
Precisely. It provides some protection against errors. If a mutation changes one base in a codon, it might still code for the same amino acid. That's pretty clever. But what if a mutation does change the amino acid?
Well, it depends on the specific change in the protein involved. Sometimes it might not have a big impact, but other times it can be detrimental, leading to a non-functional protein or even a disease.
Like sickle cell anemia.
Exactly. A single base change leads to a change in one amino acid, which drastically alters the shape and function of hemoglobin.
Wow, so even a tiny change can have massive consequences.
It can, and things can get really messy with frameshift mutations, where the insertion or deletion of 1 or 2 bases throws off the entire reading frame of the mRNA.
Like shifting all the letters in a sentence by one position, it would become gibberish.
Exactly. The order of those bases is critical for creating the correct protein.
So that brings us to translation, the actual process of building the protein based on the mRNA template.
Right. It's like a cellular assembly line following the instructions on the blueprint.
And the ribosome is the factory where this happens.
Precisely. The ribosome brings together mRNA, tRNA, and amino acids to create the polypeptide chain.
Okay, I'm ready to see how this assembly line works. What are the steps involved?
Well, it starts with initiation. The ribosome binds to the mRNA at the start codon AUG.
AUG, that's the go signal for a protein synthesis.
Exactly. And the initiator tRNA carrying the amino acid methionine binds to this codon.
Okay, then what?
Then comes elongation. The ribosome moves along the mRNA codon by codon.
Like a train moving down the track.
Exactly. As each codon is read, a tRNA with the matching anticodon brings the corresponding amino acid to the ribosome.
And those amino acids are joined together to form the polypeptide chain.
Precisely. And did you know that the formation of the peptide bond between amino acids is actually catalyzed by rRNA, not a protein enzyme?
Whoa, RNA doing the heavy lifting. That's pretty amazing.
It is. It's another hint that RNA might have been the original player in the game of life.
Okay, so the polypeptide chain keeps growing until when?
Until the ribosome encounters a stop codon on the mRNA.
A stop codon.
Yep. There are no tRNAs that recognize stop codons, so instead a release factor binds signaling the end of protein synthesis.
And the completed polypeptide chain is released, ready to fold into its functional shape.
Exactly. It's a remarkable process, don't you think? From DNA to RNA to protein, it's a symphony of molecular interactions.
Absolutely mind-blowing. But I'm guessing cells don't just churn out proteins nonstop. There must be some control over when and which genes are expressed.
You're absolutely right. Regulation of gene expression is crucial. It ensures that cells produce only the proteins they need at the right time.
So how does this regulation work?
Well, it can happen at many levels, from controlling the transcription of DNA into RNA to regulating the translation of mRNA into protein.
Okay, let's focus on transcriptional regulation for now. Can you give me an example of how that works?
Sure. A classic example is the lac operon in bacteria.
The lac operon. What's that?
An operon is a group of genes that are transcribed together as a single unit.
Okay.
The lac operon contains genes involved in the metabolism of lactose, sugar found in milk.
So these genes are needed to break down lactose.
Exactly. And the lac operon is only switched on when lactose is present in the environment.
Why is that?
It's a way for the cell to conserve energy. If there's no lactose around, there's no need to waste energy making proteins to break it down.
That's pretty efficient. But how does the cell know when to turn the operon on or off?
It's all thanks to a regulatory gene called the repressor gene.
A repressor. So it's like a switch that can turn off the operon.
Exactly. The repressor gene produces a protein that binds to the operator region of the operon, blocking RNA polymerase from transcribing the genes.
So the repressor protein is like a bouncer guarding the entrance to the operon.
I like that analogy. And when lactose is present, it binds to the repressor protein, changing its shape.
So lactose is like a VIP pass that allows the genes to be expressed.
Precisely. With the repressor out of the way, RNA polymerase can access the promoter and transcribe the genes.
Wow, it's a really sophisticated feedback system.
It is. And this type of regulation where a repressor protein blocks transcription is called negative regulation.
Negative regulation, got it.
Yeah.
So there's a whole other world of gene regulation out there.
There is, and we'll delve into that and much more in our next deep dive.
Okay, I think we need a moment to process all of this. We've covered a lot of ground from the structure of DNA to the intricacies of gene expression.
We have. And there's still so much more to explore, like the Human Genome Project and DNA fingerprinting.
Right. We'll unravel those mysteries and see what they reveal about who we are when we come back.
Yeah.
Okay, so let's talk about the Human Genome Project. It's like scientists decided to map out our entire genetic landscape, all 3 billion base pairs of it.
Yeah, it was a massive undertaking, launching a mission to explore a whole new world.
Exactly. Imagine trying to read a book with 3 billion letters.
And not just reading it, but understanding what all those letters mean, how they're organized, and how they work together.
So it was more than just sequencing the genome. It was about making sense of all that data.
Exactly. The Human Genome Project launched in 1990, and back then we didn't even know how many genes we had.
Really? So they were going in blind.
Pretty much. Estimates range from tens of thousands to well over 100,000 genes.
Wow. Talk about a wide margin of error.
It was a huge unknown. But there was this incredible sense of excitement and possibility. Scientists from all over the world came together to work on this project.
So what were their main goals? What were they hoping to achieve?
Well, identifying all the genes in human DNA was a top priority.
Okay, makes sense.
Okay.
That's like creating a parts list for a human being.
Exactly. And once we know the parts, we can start to figure out how they work, how they interact, and how they contribute to our traits and our health.
It's like opening up the instruction manual for building a human being.
Right. And another major goal was to understand the genetic basis of diseases.
So by comparing the genomes of healthy people with those who have certain diseases, they could pinpoint the genes involved.
Precisely. And that knowledge could pave the way for new treatments and even cures.
It's like personalized medicine tailored to an individual's genetic makeup.
Exactly. And beyond medicine, the Human Genome Project also promised to shed light on human evolution.
By comparing our genome to those of other species.
Exactly. We could trace our evolutionary lineage and understand how we became the complex beings we are today.
So how do they actually go about sequencing the entire human genome? It must have been a huge technical challenge.
It was. They used a variety of techniques, including something called Sanger sequencing.
Sanger sequencing. I remember reading about that.
It involves breaking the DNA into smaller fragments and then determining the order of the bases in each fragment.
Okay. And how did they put those fragments back together?
That's where powerful computers and sophisticated algorithms come in.
So it was a combination of cutting-edge technology and human ingenuity.
Exactly. And after years of hard work, the first draft of the human genome was published in 2001, with a more complete version following in 2003.
Wow, that's incredible. So what were some of the major discoveries that came out of this project?
Well, one of the most surprising findings was that humans have far fewer genes than we initially thought.
Really? How many do we actually have?
Around 30,000, not the 80,000 to 140,000 that some scientists had predicted.
That's way less than I expected. So what's all that extra DNA doing if it's not coding for proteins?
That's a great question. We used to think that non-coding DNA was just junk DNA, but now we're learning that it plays a crucial role in regulating gene expression.
So it's not junk after all.
Not at all. It's like the control panel for our genes, determining when and where they are turned on or off.
So it's essential for orchestrating the complex dance of life.
Exactly. And another surprising discovery was the extent of genetic variation among individuals.
But I thought we were like 99.9% identical at the genetic level.
We are, but those tiny differences, those 0.1% variations, account for our individuality.
So even though we share a common blueprint, it's those subtle variations that make us unique.
Precisely. And one type of variation that's particularly important is called single nucleotide polymorphisms, or SNPs.
SNPs, what are those?
Were single letter changes in the DNA sequence that occur at specific locations.
So it's like having a slightly different spelling of a word.
Exactly. And these SNPs can influence a wide range of traits from eye color to disease risk.
Wow, so the Human Genome Project gave us this incredible tool to understand our own personalized genetic code.
It did. And the applications are vast and still expanding. In medicine, it's led to the development of personalized therapies tailored to an individual's genetic makeup.
So we can potentially screen for genetic predispositions, diagnose diseases more accurately, and develop more effective treatments?
Exactly. And the impact extends beyond medicine. It's transforming fields like forensic science, agriculture, and even our understanding of human history and evolution.
It's mind-boggling to think about how one project has had such a ripple effect across so many areas.
It's a testament to human ingenuity and the power of collaboration. And as we continue to explore the human genome, we're bound to uncover even more secrets that will revolutionize our understanding of life.
Okay, I need a minute to let all of that sink in. We've gone from the double helix of DNA to the vast expanse of the human genome. But before we wrap up this deep dive, let's zoom back in on one specific technique that harnesses the power of our unique DNA sequences for identification DNA fingerprinting.
Oh yeah, DNA fingerprinting. That's become an essential tool in forensic science, paternity testing, and even studying populations and evolution.
So DNA fingerprinting. It's like giving everyone a unique barcode based on their DNA.
Exactly. And it all comes down to those repetitive DNA sequences we talked about earlier.
The ones that make up a large chunk of our genome but don't code for proteins.
Right. Those sequences are highly variable from person to person. They're like a genetic fingerprint.
So how does it work? How do scientists actually create these DNA fingerprints?
Well, it involves a technique called southern blot hybridization.
Okay, I'm listening.
First, they cut the DNA into fragments using restriction enzymes, which act like molecular scissors.
Okay.
Then they separate those fragments by size using a process called electrophoresis.
Electrophoresis, right, where they run the DNA through a gel.
Exactly. The smaller fragments move faster than the larger ones, creating a pattern of bands.
So each person's DNA creates a unique band pattern.
Exactly. It's like a barcode. But to visualize those bands, they use a labeled probe.
A probe. What's...
It's a complementary DNA sequence that specifically targets the repetitive regions we're interested in.
So it binds to those specific sequences.
Right. And because the probe is labeled, it creates a visible signal wherever it binds.
So that's how they see the DNA fingerprint.
Precisely. And this technique has revolutionized forensic science.
How so?
Well, it can be used to link suspects to crime scenes, identify victims, and even exonerate innocent people who have been wrongly accused.
It's like a molecular detective uncovering the truth.
Exactly. And DNA fingerprinting is also used in paternity testing to confirm biological relationships.
So it can help settle legal disputes or even reunite families?
Right. And beyond those applications, it's also a valuable tool for studying populations and evolution.
By comparing DNA fingerprints across different groups.
Exactly. We can learn about genetic diversity, migration patterns, and even trace ancestry back thousands of years.
Wow. It's amazing how one technique can have such a broad reach.
It is. It really highlights the power of understanding our genetic code and using it to unravel the mysteries of life.
Well, I think we've officially reached the end of our deep dive into molecular bases of inheritance.
We've covered a lot of ground, from the structure of DNA to the intricacies of gene expression and the applications of DNA technology.
I feel like I've not only learned a ton, but also gained a whole new appreciation for the complexity and elegance of the molecular world.
Me too. And I hope our listeners feel the same way.
To all you Class 12 students out there, we hope this deep dive has given you the tools and the inspiration to conquer this chapter and maybe even spark a lifelong passion for biology.
Remember, this is just the beginning. Keep exploring, keep asking questions, and keep that curiosity burning bright.
Thanks for joining us on this incredible journey through the world of molecular biology.
Until next time, happy studying, year old.